let's begin then. Well, thank you very much for inviting me, letting me talk at this Giga Consciousness seminar. So I'm Lenny from the Cognition and Consciousness Imaging Group, which I omitted in the bio wrongly here in Cambridge. I'll be presenting you an excerpt from our recent work on the neuromodulatory nuclei of the brainstem and the fact that they might have a still underappreciated role in altered states of consciousness. And I wasn't sure whether the talk is open to the public, so I'm now most likely really preaching to the choir. But for those outside the neurocritical care setting, it is, of course, really, really important to study consciousness. And it's of central scientific and clinical importance as we perform 250 million anesthesias per year, yet there are still significant occurrences of intraoperative awareness and significant parts of the many millions of cases of traumatic brain injury a year go on to be in disorders of consciousness. And even in response to viral brain injury due to COVID, there seem to be some transient and also persistent occurrences of consciousness alterations. So as your group, the GIGA, of course, knows better than anyone probably, we need to systematically understand and map what the fundamental underpinnings of normal consciousness versus perturbed consciousness are to be able to diagnose, prognose, and treat effectively in this context. And central for the ability to better diagnose and understand alterations of consciousness has been the development of frameworks such as that by your own Professor Lares that has differentiated the two main dimensions that underpin consciousness, namely the contents of consciousness, also referred to as awareness, and the level of consciousness, also referred to as arousal. And these two dimensions become disentangled in disorders of consciousness and have been associated in multiple different ways with various macroscopic neuroimaging markers. But some have actually suggested that these two dimensions can be heavily dichotomized as the cortex being responsible for awareness and the brainstem just being responsible for some sort of activating arousal signal. And I want to submit to you that such a dichotomization actually really unproductively oversimplifies the role of the brainstem for consciousness and underappreciates a key role for brainstem cortical interplay that may be key for those large-scale biomarkers many of us might be familiar with in the neuroimaging of disorders of consciousness. And I promise this is the last slide of your own amazing work. So macroscopic neuroimaging biomarkers have been critical in mapping and differentiating clinical subtypes of DSC and to step-by-step characterize the neural basis of disrupted consciousness, such as the brain-wide hypermetabolism in DSC that has been characterized with FDG-PET, structural damage associated with these conditions as resolved with DTI, as well as the crucial possibility to use task-based fMRI paradigms, such as the tennis or spatial navigation task, to detect covert consciousness. But probably the most widespread and easiest, and maybe also most clinically feasible technique remains resting state fMRI, which has converged on the importance of various large-scale macroscopic networks, particularly the default mode and frontoparietal network. And both of these disintegrate in anesthesia and disorders of consciousness, and they also alter their interplay with one another. And as a DOC biomarker, they've crucially both shown diagnostic and prognostic promise. Regrettably, however, these macroscopic network level biomarkers do not provide us direct information about potential treatment targets for conditions such as DOC, as what underpins the large scale network dysfunctions is still quite unresolved, really. So what is needed are neuroimaging biomarkers that can translationally bridge this gap. And we submit to you that using fMRI off the brainstem is in a unique position to get us closer to biomarkers that are useful to uncover fundamental mechanisms and related therapeutic interventions. And that's because of the following, because ultimately we want to understand consciousness across macroscopic and microscopic scales, like anything in biology, really. Now, while microscopic aspects continue to be well characterized using human and also animal neuroimaging, much of the microscopic aspects is tended to only in preclinical animal work. And these preclinical contexts are typically those that generate treatment targets that then enter into the long-lasting clinical pipeline. But there's overall a problem of translating from preclinical to clinical realms. But one area that holds particular promise for tightening this translational interface are the neurotransmitter and neuromodulatory systems of the brainstem. The reason for this is that the neuromodulatory nuclei of the brainstem can be addressed both in preclinical models and in humans using complementary tools. And our central hypothesis is that consciousness and the associated macroscopic network level markers need to be understood with an appreciation of brainstem mediated neuromodulation. And that these can, as a result, be a proper translational bridge that can operationalize things that are both diagnostically and therapeutically relevant. And that's because the neuromodulatory nuclei from the brainstem project globally throughout the brain and diffusely release their transmitter to change intrinsic electrochemical and synaptic properties of neurons by signaling via metabotropic receptors. And that changes the input-output relationships of single neurons, microcircuits, and of the relationship between networks. And that means that many different output types, many different architectures can be orchestrated on one and the same structural underpinning by changing their functional configuration through neuromodulation. And I think you're already hearing this, but this is logically then something that underpins large-scale network signatures, and is also the reason why we don't carry around a London bus on our shoulders, but get to reuse the same circuitries with different neuromodulatory environments for different tasks and conditions. And as a result, they're then very, very important. Neuromodulatory inputs from the brainstem are very far away from oversimplifications, such as being a lizard brain, as MacLean called them in the 1960s, that just, the brainstem just governs fight or flight, couldn't be further from the truth, because it contains all of these diverse neuromodulatory nuclei that provide the non-classical transmitters such as dopamine, serotonin, noradrenaline, and acetylcholine throughout the whole brain. And very intriguingly, recently, people have figured out that actually the monoaminergic brainstem nuclei in particular, specifically the dopaminergic source nucleus in the ventral tegmental area, are actually the brain areas that are the most strongly connected to the default mode network and frontoparietal network. So those networks that we heard are very, very heavily disrupted in disorders of consciousness. So could it be that their disruption is actually down to a modulatory deficit? And we, in fact, have known for a long time that structural brainstem damage is associated with disorders of consciousness. And this is a finding that is more than 100 years old, with the first descriptions by Konstantin von Economo, and is continuously replicated and further resolved, as in these two reasonably recent studies by Snyder and Fisher that have associated various areas in the brainstem as being coma-causing or associated with loss of consciousness during TBI. But importantly, in anesthesia, some people have reported reversible changes in connectivity of the brainstem to the cortex that, however, after they recover from anesthesia, are gone again. So could it be that some of the deficits in disorders of consciousness are not just down to structural deficits in the brainstem, but rather derived from functional deficits that we may be able to act upon? So our overarching question for this research was, could a neuromodulatory nucleus function be affected in consciousness alteration? And could in vivo functional connectivity of the brainstem, as assessed using fMRI, provide translational bridges between preclinical and clinical work? And the candidate nucleus, like I was just hinting at to look into, is the ventral tegmental area of the brainstem, the dopaminergic source nucleus. And that is because it is actually heavily implicated in parallel in preclinical and clinical contexts. Because in mice in whom the ventral tegmental area has been genetically ablated, they are found to be behaviorally unconscious until ventral tegmental area function is reinstated, in the fact that optogenetic and pharmacological VTA stimulation acutely arouse mice and can even wake them up from full anesthesia, both electrical and non-invasive stimulation. And lesions to this source nucleus actually lengthen recovery from anesthesia. So we have a very clear implication of this neuromodulatory system that acts globally in relevance for wakefulness in preclinical animals. And in parallel, in the clinical disorders of consciousness world, where we're used to looking at these large-scale network biomarkers, we actually know that dopaminergic drugs, such as amantadine and apomorphine, and quite a few other ones, have shown promising effects in treatment. And very recently, people even found a presynaptic dopamine release deficit from the ventral tegmental area in disorders of consciousness. So again, we have dopamine as being center stage for disorders of consciousness treatment. Nevertheless, no one had looked into the fact whether in both pharmacological and pathological consciousness perturbation, there may be something that it shows us that VTA function is impaired, because ultimately this could very much provide a translational framework that we're thus far missing for understanding the effects of these drugs. And the way that we approached this was to perform functional connectivity analysis of the VTA in one cohort of participants undergoing stepwise propofol sedation and another cohort of 22 disorders of consciousness patients. And both datasets had been screened for the absence of any brainstem damage so that we could get to an idea of whether there is a functional impairment universal to the two. And we extracted functional connectivity from unsmooth volumes, given how small these brainstem areas are, and then performed C2 connectivity in various contrasts, wanted to see whether a potential impairment might be related to behavioral manifestation and whether it is also related to those large-scale network dysfunctions that are so prevalent in how we look at disorders of consciousness. So just to repeat our overarching question, we wanted to know whether there is a dopaminergic parallel between sedation and disorders of consciousness that converges on the ventral tegmental area, and whether that may be a feasible mechanism for these large-scale network dysfunctions. And in order to figure out whether there is this, we started in the cohort that was being put through propofol sedation. And we first looked in control conditions and saw that the ventral tegmental area was actually strongly connected to the precuneus and posterior cingulate cortex in the control conditions, so whilst people were still awake. And already at mild sedation, we found a significant disconnection from exactly that posterior cortical region, which is a main node of the default mode network. And we found an even stronger disconnection in moderate sedation, and importantly found that as people were recovering from anesthesia, there was an increase in connectivity to this area. So we find a stepwise, reversible, and seemingly also dose-dependent disconnection of the ventral tegmental area from the precuneus and posterior cingulate cortex. And that's quite intriguing because it suggests that potentially looking at the connectivity of this nucleus might be showing us a potential dopaminergic deficit, but may also present a pharmacodynamic and diagnostic biomarker, given that it is dose dependent. And we ran various active controls of other brainstem nuclei, but didn't find any similar results. So this is very much VTA specific, dopaminergic system specific. And strikingly, we found that in disorders of consciousness patients, there was a disconnection from a very similar area of the precuneus and posterior cingulate cortex. And I'm sure you can appreciate this wonderful, massive overlap that we found there, which is really just a few voxels, but it is an overlap. And as such, we find that there's also this dopaminergic connectivity deficit in the DOC patients. So across pharmacological and pathological consciousness perturbation, the VTA, the dopaminergic source nucleus, disconnects from a main default mode network node. Quite intriguing, again, given that it is the area that is usually the most strongly associated with the default mode network, the most strongly connected. And we then wanted to know whether there's maybe a behavioral manifestation that this is associated with. And we found that the propofol participants, the people that were being sedated, they had been performing various tasks whilst in the scanner at the various sedation levels. And they were presented with verbal stimuli that they were, after they had recovered from sedation, asked to sort into familiar and unfamiliar stimuli. So that gave us the chance to use a sensitivity measure known as D prime to approximate how consciously people were able to recall given stimuli. And very intriguingly, the higher the connectivity of the VTA to that posterior DMN node remained, the better people were at recalling these stimuli. So it means that potentially there's a rate limiting function in just how conscious a given person was during a given scan. And whilst, as you probably know better than anyone at the GIGA, it is a lot more difficult to find any meaningful behavioral measures in disorders of consciousness patients, we took a longitudinal view in them and found that at a 6-month follow-up, only those patients who improved and remained alive had positive VTA precuneus connectivity reemerge, whereas those that maintained negative or further deteriorated did not improve and actually subsequently all deceased. So again, there is the possibility that this dopaminergic modulatory connectivity has a direct relevance for conscious state. And of course, the elephant in the room then ultimately is, does this connectivity have something to do with how much the default mode network, that key large-scale network, disintegrates? And the way we looked at this was to take contrasts between awake and moderate sedation in the case of the sedation dataset, and to see how this area of the precuneus it disconnects from might connect to other areas it is not typically associated with, and found that, again, the specificity of precuneal connectivity went down the more disconnected the VTA became from the precuneus. And in the case of disorders of consciousness patients, very strikingly, the more the VTA became disconnected from this posterior network node, the more the default mode network fully disintegrated in terms of losses of connectivity, not non-specificity as in sedation, but in both cases, we see a putative neuromodulatory relationship here. And that's very important because thus far, we have been missing any feasible direct mechanisms for what might underpin these large-scale network dysfunctions. And dopaminergic disconnection might be one such mechanism. And as a final last view, we wanted to look into whether this biomarker that is potentially diagnostic might have some prognostic value, as we saw in the DOC patients, and is pharmacodynamic as far as we saw in the propofol dataset, whether it can actually be upregulated by a dopaminergic agonist. And we unfortunately only had an unrelated traumatic brain injury dataset of people who did not have disorders of consciousness who received methylphenidate. But we did find that VTA connectivity to this posterior network node could be significantly upregulated by a dopaminergic agonist. And that, again, recapitulates a potential pharmacodynamic modifiable biomarker. And that ultimately tops off the fact that what we might be finding here is a key biomarker to maybe guide some therapeutic interventions in DOC and to track the effects that treatments are having. And in fact, without being too cheeky, I'm aware that maybe Leandro Sanz is present. It would be very interesting to know how a proper dopaminergic agonist, a D2 agonist like apomorphine, might fare in disorders of consciousness with regards to this connectivity. And so just again, to recapitulate, by finding this parallel between sedation and disorders of consciousness, of in both cases observing a disconnection of the dopaminergic source nucleus from a posterior network node, which is in both cases associated with behavior, outcome, and default mode network integrity, and is modifiable by dopaminergic agonist. What we might be seeing here is a biomarker for modulatory disruption of large-scale network function. And because it occurs across both, this may be something that is very much consciousness specific. Again, we didn't observe this for any of the other nuclei. And in terms of the context in the literature, this seems very, very much feasible because there are lots of structural connections between the VTA and the precuneus and posterior cingulate. And we actually know that D2 receptor, so dopamine D2 receptor binding in that region is related to default mode network integrity. And when you combine that with the fact that we know that dopaminergic tone, so overall dopamine provision throughout the brain, is globally lowered in both anesthesia and TBI models in the preclinic, that means that we may have a strong neuromodulatory consequence there. Now, of course, we will need to validate that with simultaneous PET and resting state fMRI. But given the fact that in that cortical region, we have the greatest structure function divergence, we need its concerted neuromodulation. And in fact, the precuneus and posterior cingulate cortex have experienced the greatest cytoarchitectonic expansion in human evolution, which might be reflective of the fact that they also have the most complex receptorome, so the most complex neuromodulatory landscape to keep them functioning normally. And this means, particularly with a view to the work that you have undertaken at the GIGA, that, precuneal function, which is so important for the balance between intero and extero inspection, could very feasibly be impaired by a dopaminergic neuromodulatory deficit. And this is a picture from some work that a lab colleague, Diane Liu, has been performing, differentiating the dorsal and ventral precuneus's role in intero and extero inspection. And intriguingly, the disconnection we find in the sedation participants fits exactly S-shaped between these two areas. So there may be some sort of bridge here. But anyway, this is all just to say that the precuneus and posterior cingulate may be particularly vulnerable to changes in dopaminergic modulation, which we might be capturing with this biomarker. And this might be of direct relevance to conscious state. So dopaminergic modulation of that posterior cortical network node. And just as an aside, it seems that Vipassana-like meditation upregulates connectivity from an area coincident with the VTA to the posterior cingulate. I just thought that given that you work on meditation as well, this might be of interest to you. And if you want to see more about this, you can look into our paper in PNAS. But what we then have overall is a potential translational bridge between lots of preclinical findings on dopamine and those ideas about large-scale network dysfunction in DOC, and a potential way of operationalizing this into treatment contexts. But when you talk about dopamine in the context of any treatment, of course, everybody wants to bring up the anterior forebrain mesocircuit that Nico Schiff came up with in 2010. So could it be that there's also a dopaminergic modulatory deficit affecting that? And, you know, in case that some are not familiar with the anterior forebrain mesocircuit, it basically postulates that that one of the most common pathophysiological features of DOC is a large-scale loss of activity and metabolism across the whole cerebrum. So, and this is thought to be brought about by broad-scale disfacilitation, so overall loss of excitatory activity throughout the brain, particularly in the neocortex striatum, and most importantly, the thalamus. And this is thought to come about by differentiation. So as I'm showing here in red, that brings about a situation in which the striatum cannot properly inhibit the pallidum anymore. And when the pallidum isn't appropriately inhibited, it will in turn over-inhibit the thalamus, which causes broad-scale reductions in thalamic outflow, which overall disfacilitates the brain. And this enters into a sort of vicious cycle where a lack of thalamic outflow into the cortex precipitates, again, a lack of inhibition of the pallidum, and so on. The key point here is, though, that the thalamus is very much the rate limiter, as it is about its outflow ultimately. But what is underappreciated is that actually the thalamus is the main target throughout the whole brain of dopaminergic neuromodulation. And the striatum also to a slightly more secondary extent. And given that most of that dopamine, again, comes from the ventral tegmental area, and we know that it can change input-output relationships, this may, shall I just quickly. I think only you can mute someone, Benedetta. Okay, cool. Yeah, no worries. But anyway, the idea is it could very well be that the change of input-output relationships in the thalamus via modulatory projections from the VTA may again be an active player in this anterior forebrain mesocircuit, in which this dopaminergic source nucleus is typically not included. So, and as I said just before, you know, Esteban Friedman and Nico Schiff actually performed a rather elegant experiment with various bolus administrations of drugs and a 11-raclopride PET to figure out that there is a presynaptic dopamine release deficit in the central thalamus. So again, given that this is the rate limiter, could it be that we can capture something similar using the non-invasive, non-radioactive measure of fMRI-based VTA connectivity? And what we found is actually that even though in seed to voxel-based analyses, as I had shown you beforehand for the precuneal disconnections, we could not find a disconnection from the thalamus. But when we took a ROI to ROI-based approach, we actually did find that controls had significantly higher connectivity of VTA to the thalamus than disorders of consciousness patients. And the fact that we were not able to capture this in C2-voxel-based analyses is probably that there's a subsample of the patients that maintain levels that are above the control mean. And this is something that I will come back to in a little bit. But overall, it's certainly the case that there is an overall VTA thalamus disconnection in disorders of consciousness as well, in addition to the VTA disconnection from the precuneus. And again, we found a potential neuromodulatory relationship between VTA disconnection from the thalamus and how the thalamus can remain whole brain connected, again, which again suggests that there might be a role to play for VTA thalamus modulation in rate limiting how well the thalamus can remain connected to the rest of the brain. And just as a secondary proof, we also found that this equally applies for the thalamus's interplay just with the precuneus and posterior cingulate cortex. But in order to have some proxy measure of thalamic outflow, we thought that maybe a large-scale complexity measure, such as effort to compress, might be good. And we ran some causal mediation analyses to figure out whether VTA precuneus connectivity or VTA thalamus connectivity might have something to do with how much there is actually going on in the brain as approximated using effort to compress. And we actually found that by far the strongest predictor, an extremely significant predictor with a direct effect, was the level of VTA thalamus connectivity. Nothing else, everything else was indirect. And this suggests that there might be a real rate limiting function for how connected the VTA remains to the thalamus, and as a result, how dopaminergically modulated the thalamus can remain. And this is, in terms of the relevance for the anterior forebrain mesocircuit, is captured maybe best in the fact that those patients that showed tennis positivity, so that responded in the fMRI scanner to the prompt of imagine yourself playing tennis, and showed motor area activation, actually had significantly higher VTA to thalamus connectivity preserved. And this is exactly this group that I had shown you beforehand, that was likely masking the effect in seed to voxel-based analyses. And it's important to appreciate that this goes beyond the function that we found for VTA precuneus connectivity, because that did not have any relationship with tennis positivity, whereas this does. So could it be that these two aspects are acting together to attune function within a component of the mesocircuit? And again, as a positive control, we ran lots of other ROI-based analyses, and this effect was not found for any of the other mesocircuit ROIs, be they cortical or subcortical. And I think this is quite encouraging for the fact that this might actually be something specific to dopaminergic modulation of the thalamus. And again, it could be that what we're capturing here is a non-invasive biomarker of this dopaminergic release deficit. And in fact, we know that in this case, structural rather than functional, but structural connectivity from the pons to the thalamus is the most predictive connectivity for recovery after DOC. And in fact, the thalamic hypermetabolism that I was talking about beforehand, that has been measured using FDG PET, is the strongest in the D2 richest areas of the central thalamus, so the D2 dopamine receptor. So again, this really speaks to such a modulatory deficit that we might be capturing here. And this might actually suggest that apomorphine, as you are trialing currently at the GIGA, may act also via direct thalamic and potential VTA effects in the mesocircuit to elicit its consciousness promoting properties. Because overall, actually, dopaminergic modulation of the thalamus in preclinical studies has been found to be very much involved in attuning the whole brain balance between facilitation and disfacilitation. So it very much seems like including this modulatory relationship explicitly might be a way to operationalizing good working hypotheses in this context. And again, very much like the precuneus, the thalamus might be very vulnerable to changes in dopaminergic modulation with a potential rate limiter function for consciousness via its function in the anterior forebrain mesocircuit. And that means that we basically have, both with the VTA's interplay with the precuneus and with the thalamus, two direct translational bridges to these large-scale network dysfunctions that we typically find. So I'm just submitting to you, this is very much just working hypotheses that I want to explore with you. Is it possible that we should be extending the anterior forebrain mesocircuit to also include, I'm trying to get my pointer here, to also include the ventral tegmental area as an active player, both of modulating frontal, so anterior forebrain components of the mesocircuit, but equally so an explicit posterior forebrain component in the form of the precuneus and posterior cingulate, and to really appreciate that the fact that there is a subcortical hindbrain component that may be directly have rate limiting function for how well the rest can operate. So do we need to really include impaired dopaminergic modulation as a therapy target? Because recently, very recently, in fact, a week ago, a Chinese group of researchers demonstrated that in mice, non-invasive ultrasound stimulation of the ventral tegmental area actually wakes up mice that are fully anesthetized. So is it maybe time that we consider some brainstem targets for non-invasive, low-intensity focus ultrasound? I'm just wanting to debate that with you. I have no clear-cut opinion on it. But overall, one last thing that I just wanted to say is that, of course, dopamine isn't everything. And what we really should be looking into is to have real guidance derived maybe by personalized brainstem connectome mapping. And by that, I don't just mean functional MRI, but ideally, functional MRI and DTI to know whether there are some connectivities from the brainstem that are not structurally damaged, but just functionally aberrant, that we can upregulate with the right combination of various treatments, some stimulation, some pharmacological. But I think it's an exciting prospect that maybe we have non-invasive tools that allow us the assessment of brainstem nuclei. And of course, for this purpose, we have to still very much replicate this work. And we have various ongoing projects that are doing that in center TBI, where we see that patients very much cluster according to different transmitter deficits, and they seem to be consistent with various pre-existing genetic predispositions for transmitter systems, SNPs in transmitter system relevant genes, I should say. We also find that in psychedelic administrations, there's a very selective downregulation and disruption of serotonergic brainstem nuclei, but not of others. And we're also currently trying to replicate our anesthesia results at 7T so that people will believe that this can really be done at 3 Tesla. And finally, we're also looking into how this is being affected in coronavirus survivors in long COVID. So if I can just leave you with one central thought with all of this data and all of these considerations, it seems like functional brainstem imaging is a tool that might have clinical and translational potential. And it is unique because it can allow this translation of insights from preclinical animal studies into the clinical human realm. And I think this is a bidirectional translational potential. And we should really not underappreciate how much that can do and how that can help us come up with frameworks and biomarkers that have diagnostic, prognostic, and treatment informing value for people with DOC. And I think what that amounts to ultimately is, and maybe it can all be summarized in this one sentence, is neuromodulation is much more than mere arousal. And we really need to get to real grips with this concept in order to make proper progress towards treatments in this area and towards an understanding of consciousness that isn't too stuck up and superficial. So thank you very much. And I'd like to acknowledge all the help I've received from my supervisors, the rest of the lab, and various collaborators far and near. So thank you very much.